Hello, students. A very good afternoon. So we were talking about biomolecules, and yesterday we had a brief introduction to what are biomolecules and what is their significance. And also we were talking about micromolecules in that we spoke about amino acids especially, and we were talking about what are lipids, right? So in lipids, as I was saying in the last class, in lipids we have two important factors for the composition of lipids. So one was fatty acid and the other one was glycerol, right? So we will continue from learning simple lipids and we will see what are the next topics we have today. Okay. So as I was saying, when we look at a fatty acid chain, right, or when we look at a lipid chain, the two important components that we have for the to make a fatty acid is the first one is glycerol, right, and the second one we have is fatty acid. So when we look at glycerol, as I said, we have three carbon atoms in glycerol, right? It is made up of three carbon atoms and it is made up of five hydrogen atoms and it is made up of three OH molecules, right? So that is called as a glycerol. Now to this first component, right, for the glycerol, when a fatty acid chain is attached, for example, let me show you. Okay. um, for example, if I take a fatty acid chain, so this is a fatty acid chain. Okay, so you have a fatty acid. What is a fatty acid? Fatty acid is a long, okay, hydrocarbon or carbon hydrogen chain. So usually the number of carbon atoms varies from 8 to 32, but usually in our natural occurrences we will have from 8 to 18 or 8 to 19 carbon atom containing fatty acids. Okay, so why do we call it as a fatty acid is because to this long chain of hydrocarbons, okay, at the last carbon you have a carboxylic group attached here. Okay, you can see here COOH, which is a carboxylic group, and that gives the acidic nature. Okay, that gives the acidic nature for your what you call fatty acid. That's why we call it as an acid. All right, so now to this glycerol, we have glycerol 1 and 2 and 3 carbon atom, isn't it? So to this glycerol atom, a fatty acid chain will come and join with it. Okay, and when it joins, now we have, okay, what we call it as monoglycerides or diglyceride or triglycerides. So this monoglycerides or diglycerides or triglycerides, what we talk about now, this is nothing but fats. Okay, so now usually what happens is when you have a linkage like this, right? So as I said, when you have an attachment of a fatty acid, okay, to a glycerol molecule, so once it attaches, now we can separate this entire fatty acids into two essential groups. So one type of fats, we call it as saturated fatty acids or saturated fats, or other type of fats, we call it as unsaturated fatty acids. Okay, so usually what is saturated and what is unsaturated fats? Okay, so we usually call them as polyunsaturated fat or polysaturated fats. Okay, it's very interesting to learn about this saturated fats or unsaturated fats. So let us talk about this saturated fats. Now what do we call as a saturated fat? So I'll give you an example here. So look at the fatty acid chain here. You can really see here there is a chain of these hydrocarbons, isn't it? If you observe this chemical structure, you can only see a single bond. So you can see hydrogen and carbon has only single bonds. So that is the first criteria. And what is the second criteria? When you have only single bonds in a chemical structure, that means that you can no more add any of the hydrogen atoms. Okay, you cannot add any extra hydrogen atom to that chemical structure. So that is called as saturated. Saturated means what? You can't add any more. So these type of fats are called as saturated fatty acids. Okay, so usually saturated fatty acids are, when you consume a saturated fatty acids which is coming from your meat source. Okay, so when you essentially eat a lot of meat, you get a lot of saturated fatty acids. And when you consume this meat source, usually saturated fatty acids can cause clogs or plaque formation in your blood vessels, which is really helping you to get a cardiovascular disease. Okay, so that is about saturated fatty acids. And coming to unsaturated fatty acids, Now, when we talk about unsaturated fatty acids, usually they have a double bond. Okay. So what is unsaturated fatty acids? You can see here in the slide. So they have a carbon-carbon double bonds can be seen in a chain of this fatty acid. So you can see here in oleic acid. So you have a double bond here. Okay. So imagine a chain of hydrocarbons like this, it will be, right? So imagine there is a double bond here, and imagine there is a double bond here. All right. So again, we take another chain of fatty acid, and let us assume that there is one double bond here and there is another double bond here. Now, this analysis is very important for us to understand again two more different categories of unsaturated fatty acids. So when we talk about unsaturated fatty acids, based on the geometric position of the double bonds, we can classify them as cis or trans. Okay. Now, what is cis or trans? Now, this we call it as cis unsaturated fatty acids, and then this we call it as trans, okay, unsaturated fatty acids. What does it mean? That means if you have the double bonds on the same plane, that we call it a cis unsaturated fatty acid. And if you have it on different plane, one below and one above, this we call it as trans unsaturated fatty acids. Now, generally you get across this term called as unsaturated fatty acids, right? For example, if you talk about cis unsaturated fatty acids, they are quite good for your health. They help you to build immunity, they help you to deposit good fat in your body. Okay, they will give you a proper insulation, so they are very essential for your body. Usually they are known to reduce your blood cholesterol levels. The cis unsaturated fatty acids. Okay, I'll just mark it here. Okay, the cis unsaturated fatty acids, what we are talking about. So these fatty acids are quite good for your health. Why? Because they reduce your blood cholesterol level, okay, and they make you more healthy. Whereas when you talk about trans unsaturated fatty acids, now usually when we talk about trans unsaturated fatty acids, they're usually artificially done. For example, vanaspati ghee that you use in your home. So what they do is for this saturated fatty acids, they usually hydrogenate it, or they pump hydrogen into the tanks. So when they pump the hydrogen in this tank, that will lead to the formation of this trans structures, that is alternating carbon atom structures, or double bond structures. So once it forms the trans structure, usually trans fatty acids are really, really harmful for your health. Usually they are known to increase your cholesterol levels in your blood, and they are more prone for the people who consume a lot of these trans fatty acids. Okay, they are more prone to get heart attacks. All right. So we talked about fatty acids. What is a fatty acid? Okay. What are fats we talk about? In fats, we have glycerol and fatty acid. When you combine glycerol and fatty acid, what do we get? We get monoglycerides, diglycerides, and triglycerides. So we talked about triglycerides, okay, which are usually called as fats. So in triglycerides, we talked about what is cis and what is trans and what is saturated fats. Okay. Now we are finishing with the fats. Do you have any doubts? And since I did not ask you a technical question, is my voice and video clear? Can you please respond in the chat box? So if you have any doubts, you can put it in the chat box and we can move on with the session. Okay. So as I said, okay, this is about this triglycerides. Okay. Now coming to the compound lipids. Okay, there is one more type of this lipids that is called as phospholipids. So usually when we talk about fats, okay, when we talked about fats, I told you fats are hydrophobic. They are non-water interacting, right? So they fear water and they do not interact well with water. For example, oil, when you put it in the water, they just float away and they don't interact with the water molecules. That's why we call it as hydrophobic. So when you look at another category of your lipids, you have one more category called as compound lipids. And the example for compound lipids is phospholipids. Okay. I'll write it here, phospholipids. Now, you might have heard phospholipids a lot. For example, when you consider your structure of the plasma membrane, we talk plasma membrane is a lipid bilayer, we call it as, right? So when we talk about the characteristics of plasma membrane, you have heard about it, that the plasma membrane has two different ends. One is a hydrophilic and other one is a hydrophobic ends you talk about, right? So we know that when we have two membranes of the plasma membrane, right? So this double membrane, usually this we'll call it as outer and this we will call it as inner membrane. Okay. So when you look at this outer and inner membrane, we know that these are water interacting, okay, or water loving, I will just write it as, and the inner membranes are water hating, right? So we know about this plasma membrane. Now, the characteristics of this plasma membrane is very important for the quality or the cell membrane itself. So why we are talking about plasma membrane here when we are talking about compound lipids? In compound lipids, one of the important lipids are phospholipids, and phospholipids are quite important in our biological system is because of their amphipathic. Okay, I'll write it here, amphipathic nature. What is amphipathic nature? That means this particular lipid molecule behaves like a amphibian. That means it is both water loving and it is also water hating. For example, if you look at the structure of lecithin, which is a phospholipid, so you have this phosphate head, isn't it? So a phosphate head, it is, for example, it is attached to the glycerol. Okay. And for example, you have C and C and C are the 3 carbon atoms of your glycerol molecule, isn't it? So to this, you have a phosphate molecule attached, and then you have, let us say, you have a fatty acid attached here. Okay. So whenever you have a fatty acid chain and a phosphate molecule is being attached, so usually their chemical structure is quite different from what we call it as phospholipids. Okay. So you can see here, fatty acid plus glycerol will give you plus phosphate groups will give you phospholipids. So when we talk about this glycerol, usually what do we call it as? We can tell that, okay, for example, if I tell glycerol, okay, for example, you can just write it like this, okay, 1,2 glycerol, okay, 1,2 glycerol monophosphate, okay, monophosphate. It chemically becomes 1,2 glycerol monophosphate or 1,3 glycerol monophosphate, something like this. So what am I telling? When you have a combination of your glycerol, okay, to glycerol, when you have a phosphate molecule and then you have your fatty acid attached, so it is essentially, it is just 1,2 glycerol monophosphate or something like this. It is not phospholipids. Later, what happens is an alcohol molecule modifies this entire structure to become phospholipids. Okay. So if I ask you what is a precursor of the phospholipids, you should tell that it is 1,2 glycerol monophosphate. Okay. So that would be the legit answer for what is the precursor of your phospholipids. So phospholipids are amphipathic, as I said. So a part of your phospholipids, they interact with water very well, and a part of your fat, phospholipids do not interact with your water molecules very well. So let us see how does it work here. Okay. For example, as I told you, if you consider the structure of your phospholipids, you have it here like carbon, carbon, and carbon, which is a glycerol, and here you have a phosphate molecule attached, and then let us say that here you have a saturated fatty acid, and here you have an unsaturated fatty acid attached to the glycerol molecule. Okay, we have 3 components you can see here. One is a fatty acid component, one is a glycerol component, and other one is a phosphate component. Now the phosphate component of your phospholipids, they are hydrophilic. Okay, hydrophilic. That hydrophilic head I will write it as, okay, they are called as hydrophilic head. That means a phosphate group of your phospholipids, they interact with water, whereas you have your fatty acids, that is saturated fatty acid, and you have unsaturated fatty acid, isn't it? So we know that these fatty acids are water hating or water fearing. So this will become the tail of your phospholipids. They become what? They become what? Hydrophobic. So usually the phospholipids are quite unique. Why? Because when you drop a phospholipid into the water, they form this micelle formation. Okay, micelles, you would have heard about what is micelles. So micelles are like this, you have a hydrophilic head and you have a hydrophobic tail. You have a hydrophilic head and you have a hydrophobic tail. So when I drop it in water, what happens is the entire outermost region like this in circle is covered by hydrophilic heads. Okay, and the inner portion of your phospholipids are covered by hydrophobic tails. So this is about phospholipids. And because of the characteristics of the phospholipids, okay, you have your, what do you say? You have your characteristic feature for your plasma membrane. Okay. I hope this is understood. And another part of your lipids is your derived lipids. And in derived lipids, you have one of the best example is sterols. Okay, so we can call it as sterols. Okay, what are sterols? So you usually come across a term called as cholesterol, right? So you always think that cholesterols are bad because they always end up in cardiovascular problems like heart attacks or something like that. Okay, but sterols are quite good for your body. And when you look at a sterol structure, it is very easy to recognize a sterol molecule. And how is it possible? If you look at a sterol molecule chemical structure, you have essentially 4 ring-like structures. Okay, you will have 4 ring-like structures, and to these 4 ring-like structures, you have a OH group attached to one of these rings as a functional groups. All right. So this structure we call it as a general structure of sterol. Okay. So why sterol is so important? Because some of the sterols are usually participating in your growth hormones. For example, testosterone. Okay, progesterone, which this is all sterol derived hormones, I would say. Okay, so there are 2 different types of steroid, cholesterol. When you talk about, you have low density lipids and you have high density lipids. So based on the characteristic of this cholesterol, it might be beneficial to health or it might be harmful to health. So now when it comes to derived lipids, we can take an example of sterols, and you know the structure of sterol, and then you can easily summarize with what sterol molecule is. Okay, fine. So is it understood about lipids? Now we finished lipids. Okay, and the different types of lipids. Okay, you can put it in the chat box if you have any doubts, since we have only 45 minutes for the session. So it's hard for me to just wait on for the doubts. Okay, just put it, we can solve it. All right. So the other essential component of your, of your biomolecules, when we talk about micromolecules, is sugar, right? Is sugar. So sugars are very simple, okay, carbon, hydrogen, okay, carbon, hydrogen, and oxygen containing molecules. Usually the carbon to hydrogen to oxygen ratio is 1:2:1. In the structure of your glucose, you can see that if I take an example of glucose, you have C6H12O6. That is, if you see the ratio of this, carbon is 1:2, hydrogen is 2:2, and again is oxygen is 1. So the sugars are, carbon, hydrogen, oxygen containing compounds. Usually they add sweetening to your food. For example, it is present in vegetables, they are present in grains, they are present in your fruits. Okay, the sweetness that you feel in your tongue, it's all because of this. So in sugars, we have simple sugars like monosaccharides, which are made up of only one type of sugar molecules. For example, glucose and ribose. If I take an example of glucose is made up of C6H12O6. And when you look at the chain of glucose, they can contain, as a monosaccharide glucose, 4 to 8 monomers will be there in a glucose molecule. Okay, so this is about simple sugars. Simple sugars are just simple compounds which are belonging to micromolecules. Okay, so they add sweetening to our the food that we eat. Okay, and they belong to micromolecules is because of their very, very small structure and very, very small number of monomers that are repeating in this structure. Okay, so this is about simple sugars being one of the examples for micromolecules. Okay, and the next important micromolecule that we have to study is nitrogenous bases. Okay, so if you look at your environment and if you are wondering like, oh, human beings are so different from any other organisms, or if you think any animal is special or any animal is very important or any organism is important on the planet, so just remember this: from your bacteria to human beings, all of us are very, very same in molecular basis. When you look at the molecular basis, all of us have the same genetic structure. So to study, to understand what is same between me and bacteria, you have to really understand what are nitrogenous bases. So when we talk about nitrogenous bases, they are essentially found in your nucleic acids, especially they are found in the composition of your DNA, and they are also found in your RNA, okay, which are very, very important for your expressions, genetic expressions. Now, what are these nitrogenous bases? So nitrogenous bases usually contain, okay, your adenine, okay, thymine, cytosine, and guanine and uracil are some of the nitrogenous bases. Okay, so I can just write it as A, T, C, G. Okay, so I'll write it here. I'll write it as A, T, C, and G and U. Okay, in a simple analogy, you can just call it as A, T, C, G, and U. So these are called as nitrogenous bases. Okay, so coming to hold on a second. If I have the slide, okay. Yeah, coming to yeah, here we have. So coming to the nitrogenous bases, as I said, we have A, T, C, G, and U. You can see here A, G, C, T, and U. And I told you nitrogenous bases are very important for what? Making up your genetic material, that is your DNA and RNA. So when we look at these nitrogenous bases, we can categorize them into two basic structures or two basic groups based on their chemical structure. So those are, the first category is called as purines and the second category is called as pyrimidines. Now, what is the difference between purines and pyrimidines? For example, if I take purines, okay, when I look at a structure of a purine, okay, what I can see is it is essentially called as heterocyclic. Okay, I'll write it as heterocyclic compounds. Hetero means different, cyclic means cyclic structures. So in heterocyclic compound, you can see this structure, right? You have one cycle here and you have another cyclic structure here. So in heterocyclic compounds, what you can understand here, one structure of this, one ring, I'll write it as 1 and 2 ring. So the first ring of this heterocyclic compound contains 4 carbon atom plus it contains 2 nitrogen atom. You can observe here. Okay, you can even count here in this structure. And again, if you see the second ring, it is made up of, okay, you have 3 carbon atom and then you have 2 nitrogen atoms. Okay, you can just count. So the first ring that is made up of 4 carbon atom and 2 nitrogen atoms, that is called as purine ring. Okay, purine ring. Okay, whereas you have your second ring, which is made up of 3 carbon atoms and then you have 2 nitrogen atoms, isn't it? That is called as imidazole ring. Okay, I'll write it again here. Imidazole, IMIDAZOL. Okay, imidazole ring. All right, so I will erase this. Wait. Yeah, I'll erase it so that you can understand. So what did I say here? In a heterocyclic compounds like purines, it is made up of 2 rings. The first ring is made up of, okay, 6 atoms where you have 4 carbon and 2 nitrogen, and this we call it as purine ring. And whereas the second ring, which is made up of 5 atoms, which contains 3 carbon and 2 nitrogen, and this ring is called as imidazole ring. So since they are heterocyclic compound, and in heterocyclic compound, purines are usually adenine and guanine. Okay, A and G are your purines. Okay, whereas when it comes to pyrimidines, you can see that they contain only 1 ring and they contain purine ring. It is made up of what? Purine ring. Okay, so you can see here 1, 2, 3, 4 carbon atom and then you have what? 2 nitrogen atoms, right? So whenever you have 4 carbon atom and then you have 2 nitrogen atoms, okay, this is called as what? This is called as, okay, just give me a second. This is called as what? This is called as purine ring, right? So fine. So now we understood. Okay, now we understood what is purines and what is pyrimidines, right? So take an example of pyrimidines. The examples of pyrimidines are cytosine, thymine, and uracil. Okay, so we understood what are nitrogenous bases. Nitrogenous bases are adenine, guanine, cytosine, thymine, and uracil. So based on their structures, we can divide them into purines and pyrimidines, and we understood what are purines and what are pyrimidines. Now, these nitrogen bases, they combine with other compounds to form your nucleotides. So when you look at the structure of DNA or when you look at the structure of RNA, all right, so if you take, for example, structure of DNA or RNA, it will contain 1, 2, and 3 important components to make one DNA. Okay, so what are those? The first one is sugar. Okay, and the second one is your nitrogen bases, and the third one is your phosphate group. Okay, So when sugar plus nitrogen bases plus phosphate combine, then you have a nucleotide. So let us understand how the nucleotide formation takes place. Okay. So usually when you look at your DNA or RNA, the type of sugar that is present in your DNA or RNA, it is called as ribose. Okay. It is called as ribose or deoxyribose. Okay. So you can see the structure of ribose here. Okay, this is the structure of ribose. Okay, usually this ribose sugar, what I told, it is pentose sugar. What is pentose sugar? It means it contains 5 carbon atoms. All right, 1, 2, 3, 4, 5. You can count here. So there are 5 carbon atoms. So usually if I take the structure of ribose or deoxyribose, I can write it as first carbon atom, second carbon atom, third carbon atom, and then I have fourth carbon atom, and here I have fifth carbon atom. All right, people. So now what happens for the formation of a nucleotide? Initially, what happens is your nitrogen base, which is adenine, guanine, cytosine, thymine, or uracil, they combine with the sugar. So when they combine with the sugar, at what carbon atom do they combine? They combine at the first carbon atom of what? Of your ribose or deoxyribose sugar. So here to the first carbon atom, you can see here your nitrogen base is attached. Here also you can see your nitrogen base is attached to the first carbon atom. Okay. When they attach to the first carbon atom, they have a bond. What type of bond is there? The bond is N-glycosidic linkage. Okay. So you have N-glycosidic linkage between a sugar and then you have your nitrogen bases at the first carbon atom to give rise to nucleoside. Okay. So that you call it as adenosine, guanosine, cytosine. Okay. So deoxyribo, adenosine, deoxyribo, guanosine. Okay. So this is called as their nucleosides. Now we have a nucleoside, isn't it? Already we have your, see, you can see here, a nitrogen base is there and already sugar is there. For this nitrogen base and then sugar, if a phosphate molecule is attached, see here people, you have a structure here, isn't it? So you have your sugar, which is a pentose sugar, and I told you to the first carbon atom, you have your nitrogen bases combined through what linkage I told you? N-glycosidic linkage, isn't it? Okay, N-glycosidic linkage, right? So look at here. When you look at here, fifth carbon atom to the fifth carbon atom of your sugar, a phosphate group gets attached. So when to the fifth carbon atom, when a phosphate group is attached, through what bond do they attach? Through ester bond. Okay. So usually we call it as phosphoester linkage. Okay. So to the first carbon atom, you have your nitrogen base, and to the fifth carbon atom, you have your phosphate group linked. Once that is linked, then you have your one nucleotide formation will take place. So usually we call it as deoxyribo, okay, adenosine monophosphate, deoxyribo gynosine monophosphate, deoxythymidine monophosphate, deoxycytosine monophosphate. Okay, so this is called as nucleotide. Okay, and then when you have your nucleotides, several nucleotides combine together to form a DNA. So let us understand how the DNA, how these nucleotides help in the formation of your DNA structure. So you know what is DNA. Whenever you write a structure of DNA, you usually write it like this, right? You write it like this. Usually we have a habit of writing like this, you know, the crossed helical structure. So our DNA, if you consider the structure of DNA first, it is made up of two chains, you see. So you wrote it like this. No, this is one chain and this is another chain. So since it is made up of two different chains or two different polynucleotide chains, correct, isn't it? Poly means what? Many. Okay, polynucleotide chain, isn't it? So it takes two polynucleotide chain to form one DNA molecule. And usually what happens for the formation of one polynucleotide chain, okay, you have your, look at here, this is your one nucleotide, and this is your second nucleotide, and this is your third nucleotide. If you consider one of these structures, okay, if you consider any one of these chains, you can see that this chain is made up of a series of your different nucleotides arranged in a systematic fashion, okay, to form a polynucleotide chain. And two polynucleotide chain, okay, combine together to form a DNA molecule. Whereas when you take about RNA molecule, RNA molecules are generally single stranded, we call it as. Okay, when you come through DNA, we call it as double stranded or two stranded. We will continue discussing about DNA and the structure of DNA, okay, when we talk about macromolecules. For now, when we talk about micromolecules, all we have to remember is nucleotides, okay, and how they are and what are their characteristics, okay. Fine. So the next topic that we have to discuss, hold on, we have a chat. Okay. Black fonts are not visible in dark color. Actually, I'm using red color. Fine. I have to keep changing the ink, I think. Just give me a second. Thank you for letting me know. Probably I'll use light blue. Let's try light blue. Okay. Let's see in the slide. If it's not visible, you just can let me know again so we can try. Okay. So our next topic, okay, we have biomacromolecules. Students, is it understood right now? Did you understand all the micromolecules and the properties of micromolecules? Okay. Do you have any doubts? Okay. If you have any doubts, you can let me know. Okay. We have, I think, 5 minutes, okay, to end the session. So let us finish understanding what are biomacromolecules, and then we can stop the session. So I told you in the beginning itself, whenever you have a biomolecule which weighs more than 1,000 daltons, okay, they are called as biomacromolecules. Okay. So in biomacromolecules, you usually have very heavy, okay, their molecular density is quite heavy. So that is why we have we call them as biomacromolecules. And then in this, we have several types. One we have is proteins. Okay, hold on. No, sir, we have some doubt. Yeah, can you just let me know, JNV Mahi, what is your doubt? Can you type your doubt? We can sort it. We can clear your doubts and we can end the session maybe. Fatty acids. You want me to explain fatty acids again? Okay. Yes, fine. We'll get back to the slide. Okay. Just give me a second. But we have to do it quick, I think. Okay. Because the time constraint is very tight here. So see, when we talk about fats, okay, the fats are made up of two components. One is your glycerol. Okay. This is your glycerol molecule. And the second component is your fatty acid. See, you can see here, palmitic acid. Okay. So this glycerol molecule, you can see here, the glycerol molecule and the fatty acid, they have to combine together to give rise to what? Fats. Okay. So now what happens to this glycerol? Look here, you have a glycerol here, correct, right? So to this glycerol, to the first carbon atom, you can see here, there is one fatty acid linked together, right? So when they link together through a glycosidic linkage, now what happens is, okay, so it will form monoglycerides. If you have two fatty acids, it will form diglycerides. If you have three fatty acids, you have triglycerides. Usually we discuss about triglycerides more. Why? Because these are fats, right? So in triglycerides, as I told, you have two different types of fats, that is saturated fats and you have unsaturated fats. So in saturated fats, what do we have? For example, for your glycerol, if a fatty acid chain is present and if the carbon and hydrogen, you can see here, you have a series of carbon atoms and you have hydrogen atoms, isn't it, in the fatty acid chain? So in this, if you observe this, there are no double bonds. If there are no double bonds, we call it as saturated fatty acids. Okay. So if there is a double bond, imagine that there is a double bond here. Okay. And if the double bonds are in the same plane, you can see they're only present on the upper level. So if they're present on the upper level or in the same plane, we call it as cis fatty acids. And if the plane is different, for example, you have double bond on the up and again you have in the geometric position down or the lower position, this we call it as trans fatty acids. Okay, so we understood what is cis and what is trans fatty acids. So do you still have a doubt on the structure of fatty acids? I mean, like fats, basically. Can you let me know? I think you're quite late for the session because we discussed about it. Yeah, thank you. Anyways, students, I will end this session right now and we'll freshly start off with biomacromolecules. Okay, and we can carry on. I think it's still 4:30, isn't it? The class. Can you please confirm me? It's 3:50 to 4:30, right? Or 4:35? Yeah. So we have to talk about biomacromolecules. I think we can start it in the next session. So there are no polls. So again, I had to submit you an NPS form where I had to ask about if you can understand me well, or is the concepts delivered properly? Okay, is the class interactive or interesting? So apart from the positive feedback, if you have any problems with the sessions where you're not able to understand or where you need a repetition, or if you need anything that has to be changed in the methodology of teaching, you can please let me know and I can carry on this to the next session. Okay, is it fine? So by the time you do that, I'll just see the YouTube and check on our other students. Okay. Yes. Thank you, sir. Gopal Pandey, sir. Thank you very much. Anyways, okay, students, I'll see you tomorrow. I mean, like day after tomorrow in the next session. And I hope we can have a little bit more interaction and a little bit more content delivered. Okay. Thank you very much. We can end the session now. Bye-bye.